In the last video, we began introducing translation. Now we haven't talked about the actual steps yet, really just the different components that you need for translation to take place. And we covered tRNA. Now something you may or may not have noticed is I didn't talk about the difference between prokaryotes and eukaryotes, and there isn't. tRNA functions, works, and has the same form in both prokaryotes and eukaryotes, so they are exactly the same, and the wobble hypothesis applies to both. The next component we'll talk about before talking about the translation steps are ribosomes, which are a really key player in translation. Now, ribosomes we know is the site of protein synthesis, but a ribosome itself is made up of both proteins and rRNA. That rRNA stands for ribosomal, so ribosomal RNA, and it's still the same as all the RNA we've talked about. It's still single-stranded, it still uses uracil, and it still has that ribose sugar. It just serves a different function. In this case, it serves actually as a ribozyme. We'll discuss that a little bit more, but remember, a ribozyme is an RNA molecule that can undergo catalysis, meaning it can catalyze reactions. It can make a reaction happen. So rRNA is ribosomal RNA, and it's part of this complex in the ribosome. This image, this molecular image down here, kind of shows you those different components. So in gold, all these gold strands you see is that rRNA. And these gray blobs is representing the proteins. And tRNA is going to be coming into these ribosomes, and that's what you can see here in blue, a little bit of green, and in this pink. Now the ribosome is made up of two units. You have a large subunit, so that's this top part here. This is where the active site is. This is where you're going to have actively building proteins. This is where tRNA is coming in and those amino acids are being linked together. So this is where all the magic is happening. The goal or the point of the small subunit, which is down here, is just to hold that mRNA, because mRNA is going to come in, it's going to be at that ribosome, but you kind of need to lock it in place. And that's the point of the small subunit, just to hold everything together. Now the ribosome, in more of this animated picture, it looks very globular on the outside, but it has very specific active sites and very specific locations on the inside. And these locations are designated for tRNA molecules. So before I pull up the text, just to show you, here's that small subunit. You see there's really nothing happening there. It's just kind of hugging this mRNA that you see right here. This large subunit has 3 spots for tRNA molecules. We call those the A, P, and E sites, and I'll discuss those more in a second. And then here you have that polypeptide chain growing, or those proteins being linked together. So the first site we'll talk about the A site. The A site stands for the amino acyl tRNA site, so AA. Remember, amino acyls are tRNA molecules with their amino acid group. This is the very first step, or very first location that tRNA enters. And that should make sense. It needs to have its amino acid if it's going to do anything in this ribosome. And so it comes in, and what you can see, look at this anticodon. It's matching up to the codon that's in mRNA. mRNA says GCU. This anticodon says CGA. Then this amino acyl, it's going to lose its amino acid as it moves into the P site. Think of the P site as the peptidyl tRNA site. Peptidyl like peptide. This is where a peptide bond is forming. Remember, a peptide bond is between 2 different amino acids. So as the tRNA is moving into the P site, the amino acid that it's carrying is going to link on and form a peptide bond with this growing polypeptide chain. Again, you're seeing anticodons matching up with codons. And then finally, you have the E site, or the exit site. This tRNA has lost its amino acid, so it no longer serves any function. So the exit site just prepares it to detach from the mRNA and leave the ribosome. Then it'll go to one of those amino acyl tRNA synthases, which is that enzyme that will put an amino acid back on that tRNA molecule. So now that we've talked about the components of translation, we talked about tRNA and we talked about ribosomes, we can now talk about translation in and of itself. Now, translation has the same three categories or same three components as transcription. You first have initiation, then you have elongation, then finally termination. Same three steps in translation, because you need to initiate making that protein, you need to elongate that protein, and then you need to terminate that protein. But this first step is a little bit different in bacteria versus eukaryotes. We'll talk about bacteria first. So in the mRNA, there is a sequence that is kind of this bonding sequence. Now remember, in bacteria, this mRNA is freshly copied from DNA. There was no RNA processing. Well, part of the code of that mRNA at the beginning of mRNA was a special code. It was called the Shine-Dalgarno sequence, or in this image, it's labeled the ribosome binding site. Shine-Dalgarno were two scientists who discovered this sequence. And this sequence just says, hey, this is where you need to bind to the ribosome. This is where you need to bind to just line everything up right. And it's a little bit upstream of that start codon. The start codon is going to tell essentially the very first tRNA molecule where to put down its first amino acid. So the Shine-Dalgarno sequence, again, is on the mRNA just in bacteria, and it's a way for it to bind to the complement that's in our ribosome. Remember, ribosomes have both proteins and rRNA. So this ribosome binding site is going to bond to a section of the ribosome, an rRNA sequence, that is its complement. And that kind of anchors mRNA and positions it in the right area. Then what you see, so now we're locked in that ribosome binding site. You have your start codon centered right in the middle. And once everything's lined up correctly, then a tRNA molecule is going to come in. Now, something you may have noticed, we are still missing our large subunit. The large subunit comes in last, and that's a really key point. So the first thing that happens, mRNA comes down, latches on. It uses its Shine-Dalgarno sequence to latch on to the small subunit of the ribosome. By binding to this binding site, that's going to line up this start codon nearly right in the middle of this ribosome. The start codon is here, and that's going to attract an amino acyl tRNA, the one that is the complement to this start codon. So the start codon is AUG, its complement would be UAC. Remember, this was really the only one that I asked you guys to memorize of the different codons. AUG is our start codon, and it's always going to code for methionine. And that's what we see coming in, is a methionine. However, in bacteria, it's not a typical methionine. It's kind of been modified a little bit. And we call this modified methionine F-met, or F-methionine. And that's what you see in this image here. Essentially, it's just the methionine that's always used at the very beginning of a sequence. Why? They're not entirely sure, but they know that there is a modified methionine. They're still trying to figure out the reason why it's different. But having this methionine here, that's going to help lure in this large subunit. And you see it's automatically fitting it into this P site. And so you see everything is nice and perfectly centered. So you have AUG coding for this F-methionine, or F-met. That's going to attract in this large subunit, and it's going to essentially lock it into place. So that's how we start in bacteria. Eukaryotes really are not that different. There's just a couple small changes. So just like in our prokaryotes and our bacteria, we need to first bind that small subunit. Because remember, the small subunit, its whole job is just to hold on to mRNA. Now, in eukaryotes, we had so much processing of our RNA. This is where that 5' cap, that methylated cap on that guanine group, comes really helpful. Because that cap is what bonds to that small subunit. Remember, in bacteria, it was that Shine-Delgarno sequence. Well, for eukaryotes, it's that 5' cap that we added at the end of transcription. So that's what's going to bond to this small subunit you see here. And then, just like in bacteria, that binding location is upstream of that start codon. So now that we've kind of adjusted it and put it onto our small subunit, the small subunit is going to move that mRNA section in order to center this start codon, essentially what we saw in prokaryotes as well. And it's going to function and put that start codon right in the middle, essentially aligning it with the P site. Now, instead of an F-met, that modified methionine bonding to this small subunit, in eukaryotes, we have regular old methionine. There's nothing modified about it. So that's a key difference between prokaryotes and eukaryotes. And then just like we saw in bacteria, once you have that methionine stuck into the P site, that kind of helps attract that large subunit. So the major differences being that in prokaryotes, you have the Shine-Delgarno sequence. That's what helps mRNA bind to that small unit. Whereas in eukaryotes, you have this 5' cap. Eukaryotes also have regular methionine, whereas prokaryotes have this F-methionine, or this modified methionine. Due to time, I'm actually going to stop it right there. And then the next video, which will be the last video, we'll finish talking about translation.